Well, again, thanks for being here. I'm going to talk about some high-throughput genotyping solutions for challenges in commercial plant breeding. I'm going to start, it's a little embarrassing, but I'll start with the same concept. Where's the, oh, there it is. Wrong way. Okay, you saw this before, but I think it's important. I want to emphasize two things. One is well, first of all, this says 2x food demand increase by 2015. I think we saw 70% before. Regardless, it's a lot. So I think that's one thing to remember. The magnitude is huge. And the other thing is the urgency. It's coming fast. So we need to go big and go fast to solve this problem. And I'll show you a couple ways that we're trying to do that at Monsanto. All right, here's a specific challenge for us, and it's really around, I can state one of the solutions, of course, is genetic improvement, and the challenge is how you put all these things together. So there's lots of sources of genes that we need to combine to make crop improvement, and it doesn't matter where they come from. It's natural sources, it's biotech, it's gene editing, tons of sources. We got to use them all. But the real question is, how do you get them together? And of course, markers have become an important part of doing that. Marker selection, genomic selection, is something that my team is focused on, and implementing these sorts of processes for doing this very efficiently and in high throughput. I'm going to talk about, whoops. There's a lot of challenges to doing this, depending on what you're trying to do. I'm going to talk about two examples of challenges we face and solutions that we were able to put in place. And I should mention that these solutions require a lot of partnerships, and I want to emphasize that. And of course, here I'm going to emphasize our partnership with LGC, Douglas Scientific. That was an important relationship for solving some of these problems. We have a lot of other partners too, both internally and externally. And even as a big company, we can't solve them all ourselves. We rely on a lot of external partners to help bring solutions, and it's really important to our success. So the two challenges I want to talk about, one is this Roundup Ready to Roundup 2 yield conversions in soybean. Seems on the surface like a pretty simple challenge, but I'll show you it's a little more complex. And then the second one is around, I think, a bigger challenge, and that's implementing genome-wide selection to accelerate genetic gain. All right, so the first one. Oops, all right, so Roundup was a transformational product for our company. I think a lot of you know that. Really changed Monsanto from a chemical company into a seed and trait company. And it was transformational for farmers too. It really changed their lives, the way they farm, and a product that they loved, and really has had a lot of positive impacts on production, not just on farmers' lives, but on soil runoff and chemical usage. It has a downside, or I can say a challenge, in that we needed to continue innovating on top of this. And one way we've done that is to bring new herbicide tolerances, because our weeds continue to challenge us with overcoming Roundup resistance. But another way that we innovated on top of this was to bring a better Roundup resistance gene. And so our scientists found that if they put the gene in a different genomic location and a better haplotype, they found one that had higher yield, was associated with higher yield. And if we could swap out that second haplotype for the first one, we could demonstrate that we could increase yield by 4 bushels, which is quite a bit in soybean. I think average yields are 60 bushels or so it's a substantial amount. The challenge here was, seems like a simple problem because it's 2 single genes. And the introduction of Roundup was pretty easy because you could cross Roundup resistant by Roundup susceptible, spray it with Roundup, and everything that survived had the gene. What happened was we converted all our germplasm to Roundup 1. And so to now put in Roundup 2 and take up Roundup 1, we had to make this cross here between Roundup resistant material, all the parental lines that were Roundup 1, to this Roundup 2 source, segregate it, and in the F2, you want to select Roundup 2 but not Roundup 1. Of course, you can't spray with Roundup to do that because you can't distinguish the two genes. And only 1/16 of the plants have the Roundup 2 homozygous and not Roundup 1. The only way to do this was with markers. I think that was a big realization for our breeding staff, was that there was really something they could do no other way other than with markers. And the other challenge is that there was our whole germplasm had this Roundup 1, and we had to do this conversion. It was a huge amount of material, so we had to do millions of samples, and we had to do it with markers. And I think the other point too, we had to do this early in our pipeline. These were F2s we were doing where we have a lot of material. We wanted to just forward the stuff that had the right genes to do the yield testing later on. All right, so in order to do this, we needed to grow our genotyping capacity. And of course, we had to drop costs too, because we had to do a lot of this and needed to do it cost effectively. All right, so one solution which we've talked about a lot in the company is seed chipping. I cannot emphasize enough, though, how important this was to this particular problem. On the left there, that's how we would have done it without seed chipping. And again, tens of millions of samples, plants in the field, people punching leaf holes into racks of tubes and carrying them into the lab, drying them. And then the real hard part is going back out to the field to find the plant that you want based on the genotyping result. A lot of mistakes, in addition to being very inefficient, costly. It's ergonomically not a great job either. So seed chipping was a great solution, and we had engineers in the company that invented processes for doing this. You don't see rice there, sorry, but we do this for our large seeded crops, corn, soy, cotton. We also do a vegetable, melon, and cucumber, and then wheat. So in soy, we've done a lot of this. It's high volume, few errors, and the best part, of course, is that we have this entirely automated. We can chip seed, take the samples through genotyping. We can automatically offload seed then that has the right genotype, put it in packets, and that's what gets planted by our breeders in the field. And they can put it wherever they want it in the field. And that's great for applications where you're then going to make crosses in the field because you can plant the two parents together. So it's been a wonderful technology to really help accelerate some of these workflows. Okay, now genotyping, that was another challenge because we had to do a whole lot of genotyping. And I wanted to describe here this system from LGC Douglas Scientific that uses this array tape, and many of you may be familiar with it. So the tape is shown in the upper right there. It's a flexible tape. It's 3D 4-well format with wells depressed into the tape. The key here is that you can really use very low volume reactions into this tape, and it's very easy to handle. It winds up on spools. The machine on the left there is the Nexar. That's the reaction setup machine. You can see the tape on the spool there on the left feeds into the machine. There's a dispensing system, which is really convenient for doing high numbers of samples. You can pipette into the tape, a very low volume. And then dispensing of markers is nicely set up for doing low marker screening. And that's what we were doing here. It's really a minimum of 2 markers, Roundup 1, Roundup 2. We can add other markers too if we need to. So really very nicely set up for handling this kind of high throughput, high sample, low marker screening. This is the rest of the system, the Solex water bath on the upper left. We actually had engineers that invented water baths, or they built them, over a decade ago. And we started out with thermal block machines with a robotic arm moving plates into thermal machines, and that worked fine for a while, but it quickly became a bottleneck. And so we had engineers that built these huge water baths with robotic arms that would move plates in and out of water baths, and those worked pretty good. But these systems here, we converted completely over to these really nice design. They're energy efficient, small footprint, programmable, easy to service, have been a really nice instrument for us. So we've been very happy with the Solux pass. And then on the lower left is the assay detection machine. And of course that's needed because this is a unique tape system that has to feed through there. So we've really leveraged this quite well for this soybean conversion. Of course, we've been able to extend it into other applications too, so we use it quite broadly across a number of crops. And it's been a prime driver of our ability to drop data point costs and increase throughput, and then therefore to be able to apply this to a number of problems in plant breeding. All right. Okay, challenge number 2. And this one is so it's more complex because the trait's more complex. This is about genetic gain, mainly yield. And the real challenge here is how to implement it on a big enough scale that you can really have an impact. And as I was previously a professor at a university and did a lot of genome-wide genotyping, applying it to complex problems like yield. And we could do some great studies. And I think we advanced a lot of knowledge about this area, but we were never able to scale it to a level that you could really drive genetic gain. And that's been a challenge for the industry for a long time, is how to do that. You know, the other challenge, of course, is that we needed data that would allow us to be able to do the predictions in a way that we could really make genetic advances. And those are two things that had to come along to really make this work. But the goal here really was to shift our yield testing from the field to the lab. And you know, in doing so, we can go faster and scale up. And the challenges in doing that were that this was for us a totally new workflow. We had a whole new set of things that we had to do to make this work. And it required developing and implementing new technologies to do that. Let me just start first with a quick explanation of what genome-wide selection is. It's also called genomic selection, and there's probably other names as well, but this is a simple diagram of what we're trying to do. On the left side, we create breeding populations, and we're using genetic data to identify haplotypes and assign values to those haplotypes, trait value to those haplotypes. And we've been able to do this in the company because we've done years of this sort of haplotype trait association, so we have a pretty rich database of how haplotypes are associated with different phenotypes. So then you can, using various models, assign haplotype or trait values to new populations. So we genotype new populations, and that diagram with the bars on it just shows how you can assign positive or negative trait value on various places on the chromosome, haplotype blocks, and then you can sum those up for a particular trait on an individual, get a trait value from that. And you can do that for multiple traits, and in the end you can then base your selection on those trait values that you've estimated in a breeding population. You've seen the genetic gain equation at the bottom there. I think there's two components of this that we're really impacting through this process. One is the selection index, and that's really through cost-effective scaling. We can screen a lot more individuals through this sort of process. We're ending up selecting the same number at the end, but we can start with a lot more. So your selection index can be increased quite substantially. But the real one that impacts genetic gain is that bottom one, and that's the cycle time. So by shaving off time in our cycle process, we can dramatically increase genetic gain. And this is not the cycle to releasing a product. This is the cycle time from when you start a cross to when you recycle the parents, what you've created, back to another cross. So if you can shorten that time, you can get more cycles per unit time. If it's the same amount of gain and it's a shorter time, you're going to get overall more gain per unit time. So that's the logic behind it. But again, to do this, we had to go to a big scale in order to really cover the size of the pipelines that we needed to. Okay, so in implementing this, we realized, and we've experienced this before too, that our partner interfaces are the key to solving this, to implementing something really new like this. We had an old workflow that our breeders were used to, and I show it here as being real simple, and I saw one of our breeders in the audience, so I apologize, it's not this simple. But it was a certain workflow where they had seeds, they had to get it ready for the field, they planted it in the field, measured yield, and made their selections based on that outcome. So we wanted to replace that, at least with our early generation yield testing. Of course, we still do yield testing, don't get me wrong. What we're trying to do is replace that first generation yield testing, where the estimates from the field are pretty inaccurate. We do later generation testing where we can get more replication, but we wanted to replace this. And to do that, we had to implement this new workflow for our breeders. And a large part of this was how do we handle the samples and how do we get samples from the breeders to our lab and get them processed. So we came up with some solutions here. Some of it involved plastics, so we actually design and make our own plastics, containers that are custom built for these plastics. And then the other thing too is having a distribution system so that we can actually get these to all our locations in the world, or have them made in different locations in the world. So we got to be able to have all our partners be able to access this to put their materials into it. So they put seeds into these tubes. We usually do a bulk of a few seeds, and they'll ship them to us. And then, you know, the next challenge was grinding, and we came up with this. There's a photo there of my thumbs holding a little blue device. So that was a grinding pallet that we designed, and we actually did that through 3D printers, reiterating different designs, and then using a slow motion videotape to watch it, you know, bounce up and down in the tube to see how it was functioning, and then changing the design to optimize it. So that was another challenge that we were able to solve internally. And then, of course, then you get into the wet lab part of it, and I'll talk more about that, where we had a number of other challenges. So this is just the basic steps in the wet lab. Extract DNA. We're doing a sequencing protocol, so we have to make libraries, prepare the chips, and then sequence them. I'm just going to talk a little bit about the first part. That sample management piece, we have a huge lab that just deals with managing samples, from receipt to grinding to extraction. That's the hard part of this. And one of the elements that a solution to this, what we were challenged with was with these big tubes of ground up seeds, how we're going to extract those. And the systems we had before were really made for small tubes. So we came up with this idea of a larger robot that could manage these large tubes. So you can see the boxes of tubes on the left that go into this machine, and then the idea was out the other end we'd get into the 96-well format, which can then go on to other instruments pretty easily. We went to Douglas Scientific at the time and presented this challenge to them, and it was something that they didn't have a solution for, but thank goodness they were willing to work with us. They have some great engineers. They were willing to work with us to develop an instrument that would do this. So it's a custom robotic instrument that does this process, and I can say it was a great working relationship. We've had now this instrument working in the lab for a couple years now, and I've been quite happy with it. Once it gets into this 96-well plate, we have other robots that we put it on to clean up the DNA, and then it goes on to other processes. All right, so just to finish up, then the outcome here is that we've been able to implement this new workflow. We've got a ability now to really scale this operation and do a lot more samples to the lab than we could ever do in the field. That beaker of chips there represents the size of our North American breeding program in, I think, 2012. So it all fits in a beaker. And of course, we're able to do multiple beakers now today. So we've really been able to scale the size of that operation. And it's required then a number of innovations to be able to do that, including implementing the sample handling process that we needed for it. The output then is It's a prediction of a bunch of traits, and you can actually print it out just like a breeder notebook so that they can see rows for their inbreds, columns for their different traits. You can color code them for favorable and unfavorable. And they can, if they want, make visual selections or selections straight from this data. And of course, the nice thing about this too is that there's no missing data because we have algorithms that can predict everything, and so they get a complete data set. All right, so here's the summary. Again, we've got a growing population, a lot more people that are going to require food. We all need to move big, move fast to solve this problem. And the other thing too is that we've got to do it by partnering. And at Monsanto, we have a whole lot of people that we partner with, at least in the lab. We've got a lot of vendors and inventors for things in the lab. I've highlighted a few here that I've emphasized. My group is molecular breeding technology. We work a lot with global breeding. And then, of course, LGC Douglas Scientific has been an important partner as well. So with that, I'll thank you.